Darcy Thompson, a pioneering mathematical biologist, introduced the idea that the possible innovations which can be introduced during evolution are constrained by the inherent structure of the biological tissues as well as the physical forces exerted on them. Research in this domain works to define, implement, and then to evaluate statistical models of variability of landmark points and image volumes using concepts from shape-driven image analysis. Recently, currents were introduced in medical imaging in order to define a correspondence-free metric between curves or between surfaces. The first step after collecting a set of shapes is defining a proper shape model for further statistical analysis. A shape can be defined by a finite number of coordinate points, known as landmark points, or represented more generically as geometric distribution. The anatomical and biological structures of interest are complex and need to be characterized in a consistent, reliable manner. At the Scientific Computing and Imaging Institute, we are developing methods for representing and analyzing shapes to facilitate the study of anatomical changes. This also includes the development of shape alignment procedures, spatial correspondence estimation, and statistical analysis of shape features or trajectories. Our purpose is to characterize the neuroanatomical variations observed in neurological disorders such as autism, Alzheimer's, and dementia, to detect anomalies of the shape of the heart and its dynamic changes, to correct organ motion due to breathing and radiation treatment, and to segment axonal structures in confocal microscopy of the human retina. In neuroimaging applications, we conduct a global statistical analysis of brain anatomy to identify relevant shape deformation patterns that explain corresponding variations in clinical neuropsychological measures. The motivation here is to model the inherent relationship between anatomical shape and clinical measures and to evaluate its statistical significance. Shape analysis research at SCI brings end users another dimension of analysis for advanced imaging technologies and clinical imaging research conducted through the Utah Center for Advanced Imaging Research, other basic and clinical research efforts affiliated with the Brain Institute at the University of Utah, and collaborative research projects with the best groups across the country. The center works closely with the Scientific Computing and Imaging Institute and participates in the development of new methods and tools for image analysis, scientific computing, and visualization. Current key research topics are segmentation of magnetic resonance imaging and diffusion tensor imaging of the early developing brain in healthy and high-risk infants, longitudinal analysis of multi-shape complexes to describe growth trajectories of brain structures or to describe dynamic change patterns of the beating heart, building normative population atlases of volumetric images and the embedded shapes, and new methodologies for statistical analysis of brain white matter using diffusion tensor imaging. Biomedical applications include mosaicing and volume assembly of electron and confocal microscopy images, and the ultrastructural mapping of neural circuitry in the mammalian retina to obtain better insight into retinal diseases. The tools and methods developed through Driving Biological and Medical Applications are all open source ITK and made available to the public.